Welcome to Biology at Priyojit, my dear viewers. Today we explore a fascinating group of marine invertebrates or animals, hemichordates, which forms an important evolutionary bridge between non-chordates and chordates, just as a connecting corridor links two buildings. Hemichordates display features of both invertebrates and chordates, helping us understand the origin and evolution of chordate characters. With their unique body plan, gill slits, and stomochord, they provide key clues to the transition from simple to more complex animals. Let us study their general characteristics and evolutionary significance in a clear and systematic way. General characteristics of phylum Hemichordata. They are exclusively marine. They may be solitary or colonial, and they are tuberculous. Symmetry and germ layers. They are bilaterally symmetrical. They are triploblastic and deuterostome animals. Body structure. They have soft, fragile and vermiform body. Body is divisible into three regions: proboscis, collar and trunk. Their body wall covered by a single layered epidermis. Coelom. They are enterocoelous. They can be divided into three regions: protocoel, mesocoel and metacoel. Buccal diverticulum. They are present in proboscis. Earlier they were misidentified as a notochord. Their digestive system is complete as it consists of both mouth and anus. It is a straight or U-shaped digestive tract. Their respiration is performed by paired gill slits when they are present. They may range from 1 to numerous. Circulatory system, they are closed type, simple and well developed. They comprises a contractile heart and two longitudinal vessels and one dorsal and one ventral. It comprises the longitudinal vessels which are connected by lateral vessels and sinuses. Their blood is colorless. Their excretion occurs by a single glomerulus located in the proboscis. Nervous system is primitive in nature, mainly consists of an intraepidermal nerve plexus. Reproduction, they are primarily they perform sexual reproduction. Sexes may be separate or they may be hermaphroditic. Their gonads range from one to several pairs. Their fertilization is external. The development is indirect, involving a free swimming tornaria larva. The direct development occurs in some species. So examples: Balanoglossus, Saccoglossus, Pterobranchia, Atuberia, Rhabdopleura, Dendrographus, etc. Relationship of hemichordates with non-chordates and chordates. Hemichordates exhibit affinities with several diverse groups as outlined below. So first of all, we show the affinities of hemichordates with Nematinia or proboscis worm. Nematines resemble flatworms and possess a long protrusible proboscis. Melanoglossus shares similarities with nematines in burrowing and feeding habits. Proboscis comparison: the proboscis of Balanoglossus is compared to that of nematines. However, they similarly is superficial, as the proboscis of Balanoglossus is not protrusible like in nematines. Balanoglossus lacks the lateral nerve cord found in nematines. Affinities with Phoronida. Phoronida are worm-like tube-dwelling animals inhabiting shallow seas. Similarities between Phoronida and Hemichordates or Balanoglossus: they have buccal diverticula, which is a paired sacs from the stomach in Phoronida that resemble the buccal diverticulum of Balanoglossus. Nervous system: both groups possess intraepidermal nervous system. Both demonstrate a high capacity for regeneration. The actinotorc larva of Phoronis is comparable to the Tornaria larva of Balanoglossus. Differences between Phoronida and Hemichordata or Balanoglossus: gill slits, they are absent in Phoronida but present in Hemichordates. Nerve cord: the lateral nerve cord is present in Phoronida but absent in Hemichordates. Coelom formation: Phoronida, coelom forms by splitting of the mesoderm by means of schizocele, and Hemichordates, the coelom forms by the enterocoelic method. So conclusion: the development studies by Selys, Longchamp demonstrated that Balanoglossus cannot be grouped with Phoronida due to significant differences in their anatomy and development. Affinities with Pogonophora or beardworms. Pogonophora are marine, tube-dwelling animals with one or more anterior tentacular arms. They share several similarities with Balanoglossus, particularly in the formation of mesoderm and coelom. Similarities with Hemichordates or Balanoglossus: enterocoelomate nature. The mesoderm forms as hollow pouches derived from the endoderm. These pouches lie between the ectoderm and endoderm with their cavities forming the coelom. This process clarifies them as enterocoelomates as the mesoderm and coelom arise from the gut. Additional similarities: presence of a protocoel with an external pore. Gonads are located in the trunk. They possess an intraepidermal nervous system. Transformation of coelomic epithelium into muscle fibers and connective tissue. Differences with hemichordates or balanoclasses: central nervous system in Pogonophora, they are located in the protostome, but in case of balanoclasses, that is located in mesosoma or collar region. Tentacular apparatus in Pogonophora originates from the protosome, but in hemichordates, they originates from mesosoma or collar. Conclusion: the presence of enterocoelomate characteristics in only a few invertebrate groups suggests that Pogonophora is relatively closely related to hemichordates. Affinities with Annelida: Sprengel 1893 suggested a relationship between Balanoclasses and annelids based on the following similarities. Similarities: both body shape, both groups share a similar body structure. Proboscis and prostomium. Proboscis of Balanoclasses resembles the prostomium of earthworm. Habits: similar burrowing and feeding behavior. Nervous system: presence of a ventral nerve cord. Circulatory system: both have dorsal and ventral blood vessels. Blood flow direction: similar flow patterns in the circulatory system. Larval resemblance: trochophore larva of annelids and tornadial larva of Balanoclasses both of them share apical plate with eyes, sensory cilia and muscles. Alimentary canal differentiation like esophagus, stomach, intestine. Two anterior pairs of mesodermic vesicles, preanal ciliated ring or trochophore, which corresponds to the principal ciliated ring or tornadial nerve. Differences: dorsal nerve cord and gill slits absent in annelids. Nephridia, metameric nephridia of annelids are absent in Balanoclasses. Egg cleavage: annelids cleavage is spiral and determinate, but Balanoclasses cleavage is radial and indeterminate. Their gastrulation: annelids gastrulation is occurred by epiboly, and Balanoclasses it performs by emboly. Coelom formation: in case of annelids it is cytocilic, and in case of Balanoclasses it is enterocilic. Blastopore fate: annelid larva. Blastopore becomes the mouth, and Balanoclasses blastopore develops into anus. Primitive kidneys are present in trochophore but absent in tornadia. Preoral coelom they found in tornadia but absent in trochophore. Conclusion: the differences outweigh the similarities, indicating no definite relationship. So these groups likely evolved from a distant common ancestor, and annelid and affinities of Balanoclasses are no longer widely accepted. Affinities with Echinodermata: the affinities of Balanoclasses with echinoderms were first suggested by Metzinskov, while adult forms show minimal similarities, that is intraepidermal nervous system, so larval forms exhibit striking resemblances. Similarities between tornadial larva of Hemichordata and echinodermal larva or Bipinnaria. Both are pelagic and transparent. Early development follows a similar pattern. Their blastopore develops into the anus. Their coelom is enterocilic as in pogonophores. Coelom division: in case of Balanoclasses, there are divisions of protocoel, mesocoel and metacoel, but in case of echinoderms, they have axocoel, hydrocoel and somatocele. Hydrocoel and protocoel have external pores like proboscis pore or versus water pore. So the hard vesicle, that is Balanoclasses, resembles madreporic vesicle. Both relate to excretion. Alimentary canal division: esophagus, stomach and intestine. Ciliated bands in larva show resemblance, though considered as superficial structure. Dissimilarities between the larva of Echinoderm and Balanoclasses: Echinoderm larvae lack an apical plate with eye spots and cilia, but they have a ciliary band. But protocoel is paired in Echinoderm larvae but unpaired in Tornadia. Conclusion: the similarities between the larval forms suggest a strong evolutionary connection and a shared common ancestor of echinoderms and hemichordates. So, however, differences indicate divergence due to adaptations to distinct lifestyles. Affinities with chordata: Bettisian proposed the chordata affinities of Enteropneusta and included them in phylum Chordata based on their possession of the notochord, gill slits, dorsal tubular nerve cord, unique features of chordates. Notochord proposed similarities. Bettisian referred to the buccal diverticulum of Balanoglossus as a notochord due to their derivation from the dorsal region of the endoderm and relation to skeletal function and composition of the vacuolated cells. So, the rejection of Bacall diverticulum as notochord, Newman and others dismiss the idea for the following reasons. Developmental origin: true notochord is cut off from the roof of the archenteron, so not formed as a diverticulum, and Bacall diverticulum may not even be endodermal, as the Bacall cavity is often endoderm lined. A true notochord is a solid structure, unlike the hollow Bacall diverticulum. Position: the chordate notochord extends backward throughout the body, not just forward. Extending the notochord forward only, as in Balanoglossus, is functionally inconsistent with body movement requirements. Cellular structure: the vacuolated cells of the Bacall cavity and its diverticulum are unlike those of a true notochord. Conclusion: the Bacall diverticulum of Balanoglossus is no longer regarded as a notochord, undermining its inclusion in phylum Chordata. So, according to Newell, the notochord of Balanoglossus differs from true chordates in several ways. It lacks the sheath that surrounds the true notochord. The Bacall diverticulum has no connection with the blastopore and is not rigid enough to serve a skeletal function. The notochord lies below the central blood sinus of the proboscis, unlike in chordates, where it lies above the dorsal blood vessel. The notochords contain cavities. So, conclusion: though it shows some chordate features, Balanoglossus is closer to invertebrates and is placed in an independent invertebrate phyla by hymen. So, my dear students, we have now seen how Hemichordata, though often overlooked, occupy a crucial position in animal evolution. By exhibiting characteristics of both non-chordate and chordate, such as gill slits, a dorsal nerve cord, and a unique stomochord, they act as important connecting link that help us trace the origin of chordate features. As you prepare for university and competitive exams like CSR NET, remember that understanding their general characters and evolutionary significance is essential for grasping the broader picture of animal phylogeny. Keep revising, stay curious, and continue exploring its evolutionary story of life. Thank you, and see you in the next class.